Hi everyone, I'm going to go through the assessment for energy and ecosystems now, just to explain the answers and to ensure that you understand why the answers are correct. So you check against obviously your assessment answers, make sure you fully annotate your answers with the corrections and learn these mark scheme answers. So if we have a look at question 1, the mycorrhizae are on fungi which grow on and into the roots of plants. We know that scientists investigated the effects of these particular fungi on the growth of pea plants with a nitrate fertilizer or an ammonium fertilizer. We have seen a similar question on mycorrhiza in the booklet, so it's something that the examiner does ask about quite often. So they said the fertilizers were identical except that they contained either nitrates or ammonium ions. So the scientists took the pea seeds and sterilized their surface. They planted the seeds into soil that had been heated to 85 degrees for 2 days before. The soil was sand that contained no other mineral ions useful to the plants. Okay, so they only contained either nitrate fertilizer or the ammonium fertilizer. There was no other mineral ions in the soil that those pea seeds were grown in. So it asked why, so explain is your command word, why the scientists sterilized the surface of the seeds and grew them in soil that had been heated to 85 degrees for 2 days. So if you ever see the word sterilized, or if you ever see anything being heated at high temperatures, so 85 degrees here is a high temperature, especially for 2 days, then it's likely that we're looking for an answer to something to do with killing other microorganisms. So in this case, they're killing any fungi, any bacteria on the surface of the seeds or in the soil. So they're sterilizing the seeds to kill any fungi or bacteria growing on the surface of the seeds, and also they're heating the soil so that there's no bacteria or fungi in the soil, so that you can only see the effect of adding the fungi, not other fungi or bacteria that might be present in the soil. So it's only going to show you the effect of the added fungi, so the treatment in this case. So either the ammonium fertilizer, with the ammonium fertilizer or the nitrate fertilizer, and it's the effect of those fungi with those particular fertilizers. Part B for question 1, explain, again it's an explanation, why it is important that the soil contain no other mineral ions useful to the plant. So we have a look at the mark scheme. It says so that only the nitrate or the ammonium fertilizers affected the growth. So you only want to see again the effect of the treatments. So the treatments are presence of the fungi and the presence of the nitrate or the ammonium fertilizer. So those are the two variables. So they're what you're adding, what you're wanting to see the effect of on growth. So in this case, we don't want the soil to contain any other minerals that might affect the growth. We only want to see the effect of the fungi in the presence of these two particular fertilizers. Okay, for part C of this question, it's really important that you obviously read what the treatments were. So these are the 4 groups, the 4 treatments that the experiment was divided into. So in this case, you can see the pea plants are dividing into 4 groups. Now, group A and group C, it tells you, act as controls. So these are controls for this experiment. Now, whenever you see the word controls, you need to really make sure that your first thing you write is to show the effects of. So that needs to be the beginning of your sentence. Whatever you write down, if it's explain the reason for a control, you need to start your sentence with to show the effect of. Okay, and that's the same in this question. Now, group A and C, if you read the information, it tells you that the mycorrhizal fungi have been heat treated at 120 degrees for an hour. That means those fungi are dead. They are not alive. A fungi will not survive being heated at 120 degrees for 1 hour. And they're a control to show the effect of the nitrate or the ammonium fertilizer alone. So you can show the effect of that. So you can see the effect of those nitrates, those ammonium fertilizers. For the second mark, again, to show the effects of the fungus itself. So if you kill the fungus, and in those treatments there is no fungus, you're showing the effect of that fungus. Any question about controls, it doesn't matter what the question is, you start your sentence with to show the effect of, or so the effect can be seen, okay, of the particular treatments. The presence of the fungi in this case, or the presence of the particular fertilizer, the nitrate or the ammonium fertilizers. Okay, part D for this question, again, a very common question that you will see. So the scientist, it tells you that after 6 weeks of growth, they remove the plants from the soil, they cut the roots from the shoots, they then dried out the plant material for 90 degrees for 3 days to determine the mean dry mass of the roots and the shoots. So it asks you to suggest what the scientist should have done in order to ensure that the drying process actually removed all the water from the plant samples. So, okay, we have a look at part D for this question. So you can see that after 6 weeks, the scientists removed the plants from the soil, cut off the roots and the shoots, dry the plant material at 90 degrees for 3 days. So what the scientist was trying to find was the mean dry mass of the roots and the shoots of each of the plants. So it asks you to suggest what the scientist should have done to ensure that the water had all been removed from the plant sample. So if you have a look at the mark scheme, it's a really, really common question. So they weigh the sample at intervals during the drying process to see if the weight or the mass be even better. I don't know why the mark schemes often use the word weight. Mass would be much better. So to see if the mass becomes constant by 3 days. If the mass is constant after 3 days, then all the water has been removed. If the mass is still fluctuating, then obviously, or the mass is still going down, decreasing, then obviously not all the water has been removed. So they have to continue past 3 days. So for part E of this question, we're going to look at the conclusions that can be drawn from the data in the table. So this is a really important skill. It's an application of knowledge skill. It's very, very important that you're able to look at data and draw conclusions from that data. Now, the examiner's been actually very helpful here. They've actually given you quite a specific conclusions to draw from the data in the table. They've actually directed you at what you should be looking at. So if we put up the mark scheme here, so the first thing they've looked for is the effect of the fungus on the growth of the pea plant. So what you have to do is identify, okay, which data am I looking at? What am I actually looking at in this table? So if you're looking at the effects of the fungus, you need to compare the heat-treated fungus with the live fungus, okay, and you have to do that for both treatments with the fertilizer. So you'd be looking at comparing A with B and then comparing C with D. So if you look at the heat-treated fungus and the nitrate fertilizer compared with the living fungus and the nitrate fertilizer, you can see here and here there has been an increase in the growth of both the roots and the shoots in grams per plant. Now, if you then go on to compare the heat-treated fungus with the ammonium fertilizer and the fungus with the living fungus with the fertilizer, you can see that there is a greater growth with the nitrate fertilizer compared to the ammonium fertilizer. So that's what you would be looking at. That's the data. So you'd compare something here like you actually use the data, so 1.01 compared with 9.81. Now, if you're looking at the effects of the nitrate fertilizer and ammonium fertilizer, you're obviously going to have to look at some different data for the second part of the conclusions. So for the second part of this question, you need to look at the effects of the nitrate fertilizer and the ammonium fertilizer on the growth of the pea plants. So again, if we look at the mark scheme, they're asking you to compare the heat-treated funguses, so you're removing the effect of the actual mycorrhiza, and you are just looking at the effects of the nitrate fertilizer and the ammonium fertilizer. So you have to compare this figure with this figure and this figure with this figure. So you can see that the growth of the roots is very similar for both the nitrate and the ammonium fertilizer, and also the shoot growth is very similar. So because the standard deviations overlap, there is no significant difference between the effects of the two fertilizers. So if you look here, that the standard deviations here will overlap between we're just looking at the roots, for example, between the heat-treated fungi with the nitrate fertilizer and the heat-treated fungus with the ammonium fertilizer. So these standard deviations here will mean that these figures will overlap. So no significant difference. In part F of this question, the scientists wanted obviously to determine the dry mass of both the roots and the shoots. So it says, explain why determination of dry mass was an appropriate method to use in this investigation. So we know that the dry mass will give you a measure of the organic material within a plant, but we have to use the dry mass and not the wet mass, because we know, and we've done this in the booklet many, many times, we've explained this many times, that the water content within the plant material may vary. So if you dry the plant out, then you're removing that variability in mass produced by the water. Now for part G, it's important that you read the information at the top of the question really carefully. So it says the scientists determined the dry mass of the roots and the shoots separately. The reason that they did this was because they were interested in the ratio of the shoot to root growth in the pea plant. And it's the shoot that is most commercially important to the scientists. So they wanted to have the biggest shoot to root ratio. In other words, they wanted the shoot to be much bigger than the root. So the plant wanted to invest more of the energy in producing the shoots rather than the roots. So those would be the plants that they were most interested in commercially. So if I saw that shoot to root ratio there, what I would do is very quickly go back to my table, okay? Now, they're obviously looking at which treatment gave the best results. So We know that we would want the fungus present because the fungus obviously increases the growth. So those mycorrhizal fungi are increasing the rate of growth. So we're going to look at B and D. Okay. Now, is it the ammonium fertilizer or the nitrate fertilizer that would be would give the best results commercially? So what you have to do is you have to very quickly do a shoot to root ratio so there's your shoot, put that one first. So you take your 9.8 figure and divide that by the root biomass figure or dry mass figure there. So you're going to do 9.81 divide 1.63, and that will give you this figure of 6.09 to 1. Okay, so that's the ratio. So you've got, in other words, you're comparing these two figures, so you've got 6.09 more shoot compared to root growth here. Now, if you do the same for the ammonium containing fertilizer, so you're going to take 4.01 and divide by 0.96 to give you the ratio of shoot to root growth when there's the fungus and the ammonium fertilizer, that will give you a ratio here of 4.18 to 1. So commercially, you're getting more shoot growth with the nitrate fertilizer than you are with the ammonium fertilizer compared to the root growth. Remember, a ratio is just comparing two figures. So it says, if we look at the actual mark scheme here, you're getting the first mark for saying the fungus with the nitrate fertilizer gave larger shoot to root ratio. Therefore, it's giving the largest dry mass of the shoots. And then you'll get 1 mark for actually giving those figures for comparing those ratios. So if we move on to question 2 in the assessment, it's asking you here about farming practices, looking at pig rearing, and we're looking here at an agricultural scientist investigating the effect of temperature on pig growth and then the efficiency at which pigs are converting their food into biomass. So the first question asks you, why did scientists use the same breed with similar types of pigs in investigation? So if we look at the mark scheme, obviously we would assume that the same pigs would have, or the same breed of pigs would have similar alleles. Okay, it's not enough to say similar genes, it has to be similar alleles. Okay, because these breeds are technically all the same species, yeah? So all domesticated pigs are the same species, but there are different breeds which will have different alleles, okay? All domesticated pigs can interbreed to produce fertile offspring, but we're looking at the breed. So the breeds will have different alleles. They'll have the same genes, but different alleles, so just be careful. Why would we want to use the same breeds with similar alleles? Because they're acting as a control, aren't they? So you're removing any additional variables or any genetic factors which might have an effect on growth. So we're only therefore looking at temperature. So it's that idea again of a control, meaning that you can show the effects of one particular variable more clearly, in this case temperature. It then asks you to look at the statement that the pigs were allowed to eat as much food as they wanted. How could this have decreased the reliability of any of the conclusions drawn from the investigation? So if the pigs were allowed to eat as much as they wanted, then you expect that some pigs would eat more than others, and therefore there would be different growth rates. Also, that would then obviously affect or minimise the effect due to temperature. So it would make your results, your conclusions, less reliable. Okay, so you're looking at the effect of how much food they're eating rather than the effect of temperature on their growth rate. In the next question, part B, the examiner is asking you to look at the effects of temperature on the mean growth rate using the data in the table. So you can see here we're looking at just the mean growth rate here. So you can see that from 0 up to 20, there is a rise of the mean growth rate up to 0.85 kilograms per day. And then, so the peak is at 20 degrees, and then there is a fall. So you need to describe that rise and then fall with a peak at 20 degrees from what you can see from the data. So if that was on a graph, you'd expect to see a rise and a fall. Part I here for question B, it says a student concluded from these data that the mean growth rate of the pigs was fastest at 20 degrees. So this is really asking you to evaluate the data. So it says, do you agree with these conclusions? Obviously, you don't get a mark for no or yes ever in these questions. Essentially, you're going to have to disagree with this. So we know that the mean growth rate that was recorded was fastest at 20 degrees. But it doesn't take into account, whenever you see these, do you agree with this conclusion or evaluate the conclusions, then we don't know what's happening inside of that 20 degrees. So the fastest rate of growth might be anywhere between 10 and 30 degrees. The examiner will accept between 10 and 20 or between 20 and 30, so either side of that 20 degrees. So you're getting a mark for that. You then obviously have to explain your answer. So you need to say it would have been better if the scientist had looked at in smaller intervals. So the intervals of 10 degrees were too large. You can see that here. They should have done smaller intervals, or there should have been intermediate temperatures also tested. For the first part of C, the pigs can survive at temperatures above 35 degrees C. It says, use the data to suggest why scientists did not carry out an investigation at temperatures higher than 35 degrees. So have a look at the mark scheme. Obviously, you can see from the data in the table that growth rate is actually decreasing. So the scientists are looking for the optimal temperature for the growth rate in pigs, for that conversion of food into biomass. So it would be a waste of time and resources if they were to test temperatures higher than 35 degrees. So growth rate is decreasing from 35 degrees, we can assume, looking at the data in the table. And obviously they're looking for that high growth rate. So it would be a waste of resources for the farmer to continue testing higher temperatures than that. Okay, it does say here, ignore cruelty to pigs. That is not one of the marking points. I'm sure a pig would be relatively comfortable above 35 degrees. We don't know, but that's not one of the marking points here. For the next part, part 2, the efficiency of conversion of food to biomass was lower, obviously, at 0 degrees compared with 20 degrees. Suggest an explanation for the lower efficiency. So, in other words, the food that the pig is eating at 0 degrees, not as much of that food is being converted into the biomass. At 20 degrees, from the food that the pig is eating, more is being converted into biomass. So it's asking for an explanation, suggest an explanation for this lower efficiency. So the first statement needs to be a comparative statement. So the pigs will lose more heat at 0 degrees, therefore they will use more of that energy to maintain their body temperatures. They will lose more heat at 0 compared to 20, therefore they will use more energy to maintain their body temperature. So that's the first marking point, and says it must be a comparative statement. If you had the opposite and you said at 20 degrees they will lose less heat, so they will use less energy to maintain their body temperature, that would be fine as well. Part 2 here, it's asking, the answer it's asking for is you to say where is that heat coming from. So to maintain their body temperature, they're having to respire, that is generating heat, which is maintaining their body temperature. So you have to link the heat resulting from respiration as how they're maintaining their body temperature, and the food is obviously used, the third marking point, to fuel respiration. So they're using that food to respire. The respiration is then generating heat, that is maintaining their body temperature. So at 0 degrees, more food is required, more respiration is required to maintain their body temperature. Part D of this question is asking you to evaluate the conclusions of another part of an experiment done by scientists looking at 5 large groups of pigs. In this case, they fed the pigs a high fibre diet compared to a lower fibre diet. They're looking at, obviously it says here that high fibre diets cost less than lower fibre food for the pigs. So the graph, we look very carefully whenever we see a graph. What we're going to do first, we're going to look at the axis labels. So the y-axis says that the final mass of pigs as a percentage of the final mass of the control group. So it tells you the information that the control group was fed a low fibre food with the same energy content as the high fibre fed group. So A, B, C, D and E were all fed high fibre foods, and what we're looking at here are the bars that are representing the final mass of the pigs as a percentage compared to the control group, which was fed a low fibre food instead. So if we look at the conclusion itself, the farmer saw these results and concluded that he should replace the pig's usual food with food B. So if you're asked to evaluate, you need to give some information in support or for, or in support of the farmer's conclusions, and you have to give some against. So you're doing a for and against. You always do that in the evaluation. You also need to think about the experimental limitations of this particular example, and they tend to go in the against section, if you like. So really, the only for statement here in support of his statement is, if we look at B, you can see that food B produces a greater mass than the control, because the value there is greater, the figure is greater than 100%. So what that is saying is that the final mass of those pigs as a percentage of the final mass of the controls is greater than the control group. So food B would give a greater mass compared to the control. However, we're doing an evaluation, so you have to say, okay, what doesn't support that statement? If you look at the error bars for B, so those are 2 times the standard error, you can see that the error bar actually drops down to 100, which means that it's no better, if we take the error bars into account, that treatment B is no better than the control, and you can see that here in the mark scheme. So the error bars suggest that there's no different from the control. Also, you can see that the error bars of A and B overlap. So again, that would suggest that treatment A was better really than treatment B, and treatment B was no better than treatment A, because the error bars are overlapping. There is no significant difference between those. So you need to mention those overlapping error bars. So A is no better or no different from B. So, or the other, vice versa. So you're comparing here A and B, which again goes against the statement made by the farmer. Thinking about the limitations, well, they've given you this value for days for a reason here. They've told you it's 10 days, so is that long enough? It only ran for 10 days, so that would be an experimental limitation. Also, we know nothing about the conditions that the pigs were being kept in. Were the conditions different? So you need to think about that. So this is really digging into this experiment, what information you're being given, what information you're not being given. And then no information about the cost of the feed. So the farmer was going to swap to the feed, but perhaps feed B was more expensive. It doesn't tell you that compared to A. So why would the farmer not use A? So there's no information about the cost. So I put this question in the assessment to give you a little bit of an easy one. It is one that was in your booklet, so I hope that you've all got this 100% right. So it asks you here in question 3 to determine, that yield can be determined by measuring the dry mass. Suggest how you could determine the dry mass. So for 2 marks, what are the key things? So you're going to heat at about 100 degrees. Anything above 80 degrees would be fine here. So heat at 100 degrees, make sure the temperature is not too high, because that would obviously burn the material. So you're looking at around 100 degrees. What that's going to do is that is going to evaporate the water. And then for the second mark, you've got to mention the fact that you're going to measure the mass and then heat again, measure the mass, heat again until there's no further change in the mass. Okay, I know they use the term weigh here, but measure the mass is better, I would say, scientifically, but weigh is okay as well. For the second part here, part B, what's the advantage of using the dry mass over the fresh mass? Again, a very similar question to what we've seen before. There's lots and lots and lots of repetition in these types of exam questions, so please make sure that you do learn these mark schemes. So for part B, you just need to say that the amount or the water will vary in that plant material, and that will affect the fresh mass but not the dry mass. Okay, so we know that there's varying water contents in plant material, so we don't want that to have an effect, so we measure the dry mass instead. So the next couple of questions are looking at some maths that you might see related to this topic. Remember that across your 3 papers, you're going to have at least a minimum of 10% maths questions. So it's really important that we look at what kind of maths questions might come up. So have a look here. The first thing you need to think about is it's asking to calculate the gross primary production. So you need to think about what units will that be in. So the examiner, the learning outcome is, can you suggest a unit for GPP? So understand that unit actually helps you do this question. So it gives you information here for to calculate an area. It gives you the energy, potential energy, and it gives you a time. So your units are going to be, for GPP, are going to be, in this case, kilojoules per metre squared per year. Okay, because that's the information that you're given in the question. And we know that technically that's gross primary productivity, but the examiner is a little bit lax on exactly what wording it uses. But because it's mentioned a time in the question, you would always put that in your answer. So in this case, what you need to do is you're going to work out the area first. So 10 metres by 6 metres will give you an area of 60 metres squared. So you've got your area of 60 metres squared. So if I just bring up the mark scheme here. So that's here's going to be a 60 meters squared. And then you're going to work out how many kilojoules is in that area per year. Okay, so we know that there are 12 times 10 to the 4 kilojoules per year potentially produced by that area. So that's 120,000 down here. Okay, so if we actually convert that into normal numbers again out of standard form, that would be your figure here, okay, of 120,000. So that's 120,000 kilojoules per year that are produced by that area. So what you're going to do is to work out your gross primary productivity. You want to obviously find out how many kilojoules per meter squared. So you're going to divide the energy produced in that area in a year by the numbers of meters squared in that area to work it out per meter squared, because gross primary production is always calculated per meter squared or per hectare squared. Okay, so you're going to do 12,000 divided 60, and that will give you a figure of 2,000 kilojoules per meter squared per year. Look at your answers. Does it look sensible? Yes, it does, based on those figures. The next calculation you need to do is to calculate the energy efficiency, so that percentage transfer between each of the trophic levels in the diagram. So between producer and primary consumer, between primary consumer and secondary consumer, between secondary consumer and tertiary consumer. Really, really simple calculation, guys. So all you need to do is take your trophic level and divide it by the trophic level below. So take, in other words, for the first energy conversion from producer to primary consumer, so from the first trophic level to the second trophic level, you take the higher trophic level value, okay, so you take the value of 17,000 and divide that by 90,000. So 17,000 divide 90,000, so higher trophic level divided by the lower trophic level. You divide that 17,000 by 90,000, times it by 100 to get a percentage efficiency, and you should get the answer rounded, obviously, to one decimal place of 18.9. You repeat that for the other percentage transfer of energy. So you're going to take your 4,600 value, so that's your secondary consumer, so that's the energy found within the secondary consumer, divide that by the energy found within the primary consumer, so higher trophic level divided by the lower trophic level, and that will give you 27.1. Now notice that the energy efficiency has increased, which is what you would expect. And then tertiary consumer, so when the secondary consumer is fed upon by the tertiary consumer, the tertiary consumer eating the secondary consumer, the amount of energy the energy efficiency that you will see here will be 3,090 divided 4,600, and that should give you a figure of 67.2%. So the efficiency of energy transfer is increasing as you move through the food chain. Now think about the importance of that, or why that happens. We know that the producer will contain a lot of indigestible energy. So the producer will have lots of cellulose. The primary consumer will not be able to access that biomass, that energy stored in that indigestible parts. Also, we know that not all the plant is eaten. So we know that the roots might not be eaten. We know that not all parts of that plant might be eaten. So energy efficiency is relatively low between the producer and the primary consumer. Again, it might be even lower than 18.9%. This might be a slightly more efficient energy transfer, we know it can be very, very low, that energy transfer, usually around 10%. And that will increase as you go up the trophic levels. So question 5 is basic recall question, really, really straightforward. You need to learn this off by heart. So to describe the process of calorimetry, remember this is a way of measuring the energy stored inside organic material. So your first mark is saying, obviously I've got this straight from the booklet. So this is used to measure the chemical energy stored in organic material, so the biomass in other words. So we're describing the process. You're saying first what you're doing, so you're measuring the chemical energy stored in the biomass. Second mark is to say that the organic material has to be dried out. We already know that, so we're going to dry out, remove any of the water. The third part is that then you're going to burn in oxygen in a combustion chamber within the calorimeter. Okay, so you have to burn it in oxygen. You want it to be completely burnt. You want complete combustion of that material. Now, the energy given off is used then to heat a known volume of water. So the calorimeter, so your combustion chamber is inside a little tank of water, so that has to have a known volume. The increase in the temperature of that known volume of water will indicate the energy contained within that burnt sample of organic material. And the units, always state the units, are going to be in kilojoules that you're going to get from this calorimeter reading.